Okay, so here should be the last video. We're going to go through these two topics: how to read a paper. We're going to go through kind of first read the abstract, then the figures, then the intro. We'll go through those steps, and then how do we summarize that paper for our assignment or for our own notes. So we'll start with this paper by Stefan Devner called How About Taking a Low-Cost Small and Wireless EEG for a Walk. This is one of the most popular mobile EEG papers that came out now almost 6 years ago, but you can see it's already cited by over 200 other researchers that have been doing work in similar areas. So we're going to go through kind of how I would normally read a paper if I was perusing the literature. So of course we've already read the title by this time and we've clicked on the paper. We know some of the authors, we might know some of the work that they've done in other studies. As we get more familiar with the literature, and then we might read the abstract or start to skim the abstract. This is quite a short one, and they say that this paper is to build a low-cost, small, and wireless EEG system suitable for field recordings. We merged consumer EEG hardware with an EEG cap. Auditory oddball data were obtained, so that's the kind of type of experiment they did, while participants walked outdoors on a campus. So that's kind of the mobile setting. They did single trial P300 classification with a certain type of linear discriminant analysis to check how good their data was. They were able to classify the data both when people were inside and outside, depending on what condition of sound the person was hearing. We'll go through that. And then the last sentence is that they conclude that good quality single trial EEG data can be obtained with this affordable setup. Okay, so that's the abstract. We could even read these descriptors. Usually after the abstract, we know a lot about a paper. We know kind of what they hope to find, why they did the experiment, how they did the experiment, what they found, and then some discussion of the results. And so you can see that the abstract actually mirrors the structure of a scientific paper. A scientific paper has the introduction, and then the methods, and then the results, and then the discussion. The introduction also has kind of the literature review, as does the discussion. So this part of your assignment that you're working on now will help you with both. And you can see in the abstract that that's the same thing, that there's this introductory sentence, like what's the point of all this research? And then the methods, what they did, and then what they found that single trial P300 classification was possible, and then a discussion and a conclusion. We conclude that this is happening. And so those same sections are actually going to be present in the paper itself. Here's the introduction. Here's the short method section that ends right here. Here's the short results section, which will be easy for us to cover because it's only 2 paragraphs. And then the discussion section, which is just a couple paragraphs as well. And then this paper also has these figures. And so usually the second thing that I'll do after reading the abstract is, and sometimes even before reading the abstract, is I'll look at the figures. I'm quite a visual person, so I want to know kind of, does this data look good enough that I believe it, that I would present the data in a class or in a talk somewhere? And so I want to see how good does the data look. And so I might look at their ERPs. So here's the ERPs in these 4 different conditions. And these ERPs are being shown at many different parts of the head. And it looks like, without even reading the caption here, it looks like you can see that the blue lines are when people are inside. So blue versus blue, and the red lines are when people are outside. And that the thicker lines are when they had what's called a deviant, so a rare tone that's less often occurring that they have to count. So these participants are just listening to tones in headphones, and they have to count one of the 2 frequencies of tones. And here they're showing those rare tones are these solid lines. And what you normally see, which we've looked at in class already, is that when you're counting something, when you're paying attention to a stimulus, you get this increased positivity around 300 milliseconds after the onset of the stimulus, which is called the P3 or the P300. This seems to be related to our attention, so that the more attention that you're paying to something, the more you get this component. If you're distracted with an alternative task, usually it gets decreased a little bit, which happened in this case. That in this case, when people were outside, there was a slight decrease in the difference between the red lines compared to the blue lines. The blue line showed a bigger effect here than these red lines. And so that could be for two reasons. It could be because people were paying more attention when they were sitting inside than when they were walking outside, or it could be because of some mechanical artifact or noisy data that made the data appear in this way. And so the researchers, that's maybe one question that we might have as we're reading this paper is this an effect on the brain, or is this a kind of a noise effect in the data? What else can we see here? When we're looking at ERP plots, it's good to check how consistent they all look and how much like ERPs they all look. Do they all go up and down like the ERPs we see in our textbook? Or are they jagged and screwy lines that don't make much sense? These all look like pretty nice data. The second plot here is showing those same ERPs, but single trials. So instead of averaging over all the trials for each participant, which is what's shown here, this is the grand average across all the trials and then across all the participants. So it says grand average. This is showing a single subject, and it's showing every one of their trials. So here's when they were listening to a tone inside, trial 1, trial 2, trial 3, trial 4, trial 5, all through. The colors here are showing how big these peaks and valleys are. So you can see that, for instance, after people hear the deviant tone, which is like the blue solid line here, they get this big positivity around 300 milliseconds after the tone. And that's the P300, and this is showing it on individual trials. So you can see that it shows up on almost every trial. And you can see that both when people are inside and when they're outside. So it looks like you can tell the difference when people are inside or when they're outside of when they're hearing one of these standard boring sounds compared to when they're hearing one of these exciting sounds that was relevant for the task that they're doing. And in fact, you can get a computer to classify, which is something we'll talk about after the midterm in a great deal of detail. You can get a computer to classify, to kind of guess on any given trial, is this type of trial or this type of trial, based off how the data looks. You can train a computer, just like you can train a computer to predict other things, like what you're typing into Google. You can train the computer to predict each of these trials, whether it's from this set or this set. And here they are, individual people, showing that classification can do quite well, above 70 or 50%, both when people are inside and outside. And this is one way that we might have brain-computer interfaces in the future, that if this computer can predict just by how your brain responded to this tone versus this tone, that you were attending to this tone, the rare one, then your brain can then do some action. And now that it knows you were attending to that item, now it can engage with it, it can type that item on a keyboard. So this is kind of a promising result because it shows that with these portable, flexible EEG systems, you can measure data that's good enough to be classifiable into what type of situation it was. Okay, so those are our two figures. We might even read the figure caption just to make sure we know what we're doing here. We've got single trial amplitudes for one subject, for standards and deviants, for indoors and outdoors, and these are averaged over a couple different electrodes. You can see these lines indicate when things happen, and they are smooth in this way a little bit so that they don't look very jagged. So they average together the trials that are right after each other. And then in B, they're showing here the classification results for each subject for indoor and outdoor recording. And they remind us that chance is 50%. So if the algorithm that was trying to predict people's performance was just guessing, they should be right about half the time, whether that was a standard or a deviant. But in fact, that wasn't the case, and people were the algorithm was constantly better than chance. So it could predict people's condition that they were in, whether the stimulus was relevant to them or not, just based off the brain activity. Okay, so now if we go back to our outline, once we've looked at the figures, now we might start looking at the intro and the conclusion and then the discussion. So just like the abstract, there's some important paragraphs in the document itself. I find that this last paragraph of the intro is very important because it's the hypothesis or the thesis statement. So you'll recall from writing essays that you have to put kind of right at the end of your first paragraph what this essay is about. And same with a paper. This paragraph of the paper tells us what they're doing and why they're doing it and what they think they're going to find. So let's read it. The P300 event-related potential is a popular, flexible, and informative signal. We therefore investigated whether the single trial P3 could be reliably identified during natural behavior. To this end, auditory oddball data were obtained while participants walked around campus, and the ERP and EEG single trial data of quality were compared to a seated indoor recording condition. Okay, so you can see this is kind of like another abstract. It's another summary of what they did in the experiment, why they did it, what they did. They don't quite go into what they found because this is the intro of the paper. Okay, so after the abstract, it's good to read that last sentence of the introduction. Now, the next sentence I usually read is the last sentence of the entire paper. Where's that? The last paragraph of the paper is usually like a conclusion paragraph. And again, it's going to be really general about what this experiment was about and what it found. Various fields may benefit from wireless EEG technology. More fundamentally, mobile EEG enables the truly simultaneous acquisition of brain activity and behavior, which may be of interest to these other fields. Finally, the development of small EEG systems is necessary to achieve ubiquitous BCIs, but present evidence for successful online neural decoding is scarce. By demonstrating that low-cost mobile EEG is possible, we hope to spark enthusiasm towards more ecologically valid EEG experimentation. In the future, mobile BCIs could predict and thereby prevent maladaptive human behavior in everyday life. So now we kind of get the really big picture, and at this point I think we would be ready to write a paragraph in our summary paragraph about kind of answering these questions. What's the hypothesis? What were the methods? And what results did they find that were interesting? We've already read enough of this paper to start to answer some of those questions. I'm going to do one more video, a part 4, in which we explicitly think about these questions, and then we also go in a little bit more detail into the paragraphs of the paper that we still haven't looked at and think about what they're showing us.